with modeling gene regulatory networks, and we will look at some simple examples like abscisic acid signaling, which is a classic example of Boolean network modeling to understand a signaling network. So, let us look at more realistic examples. So, how do we model such systems, right. So, this looks at abscisic acid signaling. It is an important plant hormone which regulates stomatal closure. So, when does the stomata close, and it is dependent on a complex regulatory network that involves several different proteins and their states and so on. And so, you have some inhibitions, some activations, some complexations, so on and so forth, right. So, you have a pretty complex system. So, how do you model these? You start writing out equations like this. So, you now say that NO depends on NIA12 and NOS. So, if you can spot it in this figure, you have several nodes. So, NO is here, it depends on NIA12 and NOS, and you will see that there is a complex relationship for each of these things. So, calcium depends on calmodulin or CIS and not calcium 2 plus ATPase and so on and so forth, and ROS should not be there for something else to happen. So, you basically integrate all possible relationships into the model, and you will have to understand how each of these the representation works, and if there is the one difficult thing would be at nodes like this, where you have to find out whether it is an and or an or, right. When you have multiple inhibitions incident on a particular protein, which of them supersedes, right. So, is it an and or is it an or, right. Those things will start becoming important. So, you will have to essentially go back to the literature to find these. For example, here it says pH inhibits so many of these things, right. So, K out is pH or not ROS or not NO, K out is pH, ROS not, and NO that comes via this, right. So, it is easy to kind of frame these rules. You just pick your node of interest, look at all the incident arrows and encode them up, right, and depolar, right. So, how many arrows are incident here? 1, 2, 3, 4, right, and you have 1, 2, 3, 4 here. It's straightforward. The difficult part is making this figure, right. This involves digesting a whole lot of literature, figuring out all sorts of interdependencies and so on, and making this figure. Once you make this figure, it is easy to build this up. Let us look at another example. This is a classic example of modeling the cell cycle. So, you must be familiar with the cyclins and so on. So, there are several cyclin proteins involved and so on, and this is the regulatory logic. It says cyc D is an input and is considered constant, which means what would be the rule? There is no update, right. So, cyc D star is cyc D, right, that would be. So, RB is expressed in the absence of cyclins which inhibited by phosphorylation. It can be expressed in the presence of cyclin E or cyclin A if their inhibitory activity is blocked by P27, right. So, or cyclin E activity requires the presence of E2F and the absence of RB, right. So, it is easy to, once you write down these statements. So, these are statements that you can make from a figure like this, or rather this is what you would get from literature, right. If you consolidate, so each statement written here will have the strength, backing strength of 3 or 4 papers which try to describe these kinds of interactions and so on. So, you take these and put them together, you can then build rules such as these, right. So, cyclin D star is cyclin. So, E2F star is not Rubisco and not cyclin A and not cyclin B, or P27 and not Rubisco and I do not know if it is Rubisco, it is RB is and cyclin and not cyclin B. So, this is or let us look at cyclin E, that was the simpler rule. So, it requires the presence of E2F and the absence of RB, right. So, CDC20 is indirectly activated by cyclin B, right, and cyclin B should not have both of CDC20 and CDH1. So, cyclin B is active in the absence of both CDH20 and CDC20 and CDH1. So, you set up rules like these. So, how many states do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, what are the total number of possible states of the model? 2 to the 10, right. So, you have 2 to the 10 possible states or 1024, and you know where each of these are going to be on and off, and there are various nice ways to simulate these models. So, let us take some other arbitrary gene network, and so one thing you can do is you can just take an adjacency matrix, generate an adjacency matrix, then draw the corresponding network, write down the Boolean transfer functions for the nodes, and simulate the time course for any given node using a synchronous update. We will just see how we do this for a very simple system. We already did it for one system, but let us consider this very popular system. You can generate a random adjacency matrix, that is what I was trying to say, right. So, let us say you just start filling random numbers here, right. So, minus 0.1, 0.2, minus 0.3, 0.4, 0.8, something, right. You could have zeros also, something of this sort, and then you create the corresponding adjacency, corresponding gene network, and so on, right, and you can simulate it. Right. So, the only thing you will have to bear in mind is when you have, let us say this is C, B and A, now is D going to be B and not A, or B or not A. So, this is a problem when you start reconstructing from a matrix of this sort, but if you are going to look at literature, this will be quite obvious. It will tell you what is the dependencies. So, let us anyway leave this out for the moment. Let us come back to this circuit. Do you remember, have you seen this circuit before? It is a classic oscillator that was first synthetic, one of the most popular synthetic oscillators built. There is some sort of cyclic inhibition. There were 3 proteins used, I think they were lambda Ci, lac-I and tet-R with a GFP reporter. Right. So, you have a system like this. Now, what are the rules that you would write? A star is not C, B star is not A, C star is not B. Now, let us look at 0 0 0, this will just become 1 1 1, which will again become, so you have a nice oscillator. And the other states will again, you know, you will see that they cycle through several other states. So, let us start with 0 0 1. So, this will become 0 1 1, 0 1 0, 1 1 0, 1 0 0, 1 0 1, 0 0 1. So, here again you have 2 cycle attractors.
What do you mean by you can construct, you will not know B and not A or B or not A?
No, I mean if you just had an adjacency matrix kind of representation, you would not know, right. So, I have a node like this D which has 2 incident nodes and a repression from A and then activation from B. Which of these supersedes? So, is it B and not A or B or not A. How important are these two interactions relatively?
So how can we tell that from graph?
You can't tell that from a graph. So, you can't tell that from an adjacency matrix either, right. So, the best way to go about this is you look at the rules carefully and you construct the Boolean transfer functions appropriately and then simulate them. There are nice tools that are there for simulating these. I will show them in the next lab. So, another example system of a example of a discrete system is this Conway's game of life. I do not know if you are, some of you are familiar with this. It is this you can think of a very large grid and you start with some sort of pattern like this, saying there are bacterial cells present here, and so at each time you do an update. So, every given square has 9 adjacent squares. If at least 5 of them have bacterial cells, then this will become on, else it will go off. So, this is also like a classic cellular automaton. Right. So, then in the next step here you will see that this has so many neighbors active. So, this will again remain on, but many of this will go off. So, it might be you can say at least 3 need to be on or 5 need to be on and so on. Depending on that you will get different patterns and so on. So, you can just update it, but basically this is a discrete update, right. So, your x of t plus 1 depends on x of, or let us say x of I comma j depends on x of I minus 1 j I j minus 1 I minus 1 j minus 1 I plus 1 j I plus 1 j plus 1 so on, right. So, all these are going to affect each state, but this is just a nice example for how you can do discrete modeling, right. Most of the modeling that we have seen so far are in some sense a little more continuous. Of course, networks are discrete, the initial networks that we saw in the first module of the course, right. So, the good thing is you can use this kind of Boolean models for very complex scenarios, right. You can use them. So, let us maybe go back a couple of slides. So, let us just consider this rule. Here the level of P27 could very well come out of some other signaling network or something like that, which you may obtain by solving ODEs and then maybe do a simple thresholding to say whether it is on or off. So, you can use this these kinds of Boolean models as a sort of framework to which multiple other kinds of models can connect. So, this the level of cyclin B could come from an FBA flux if you want, right. So, you can do things like that. So, you can use these kinds of discrete models to connect several models. So, there are very nice software tools, the best of them being the more recent BoolSim, which we will work on in the lab in the next class. So, this is just a JavaScript based simulation which is just so easy to use. You literally copy paste a bunch of rules like A star equals not B, B star equals not C and click simulate, you will get a nice time course. This Boolean net which is based on Python, again very useful, very powerful. It can automatically do things like knocking out a single node. As always we want to do some perturbations and it is quite easy to do perturbations using tools like Boolean net. Right. So, what does perturbing here mean?
Changing the rules.
Or what does deleting a node mean?
I think we have to remove that from every rule.
So, make it permanently false, right. So, make a node permanently false, right. So, if the node is permanently false then it is going to have an effect. So, you just make the update rule for that node as A star equals false.
So, what if that node is present in some other?
It will just be false because it is that protein or whatever is no longer present. There is also meta-reg which uses heuristics to identify steady states and consistent regulation functions and so on. So, there are many interesting tools that are available. So, in this video we looked at some examples, and I hope with these examples you have a good understanding of how one builds and simulates models of Boolean networks to understand, say, signaling or gene regulation. In the next video we will look at continuous models, wherein we look at models similar to the dynamic models we saw much earlier on in the course for gene regulation and so on.